review cellular respiration with you guys. So first I'm going to start with glycolysis. Glycolysis means to split sugar. So we're going to start with our 6-carbon sugar molecule. So each one of these circles represents a carbon. So we start with glucose, which has 6 carbons. Of course, there's hydrogen and oxygens associated with each of the carbon, but in this class, I just want you focusing on the carbons and ATP and the electron carriers. So let's start with glycolysis, which occurs in the cytoplasm. So in the cytoplasm of the cell, we have, like I said, 6 carbon molecules. The first stop is to put a phosphate group on there. So we kind of talked about phosphorylating. This is substrate level. Phosphorylation. So this is where, with the aid of an enzyme, it is going to go ahead and take a molecule of ATP and transfer that phosphate on to glucose. Alright, so that's the first step. Once we have done that, it also ensures that glucose cannot leave the cell. So we have our glucose 6-phosphate, and there are other steps that are going to take place, but again, for this class, we're kind of summarizing this down. So what we're going to do is we're going to repeat this process and stick another phosphate molecule onto our glucose. So we need another molecule, ATP. So far we have invested or used 2 molecules of ATP in order to get this chemical reaction to take place. So we have our, as remember, we had our OOP and now our big poop here. And now what we're going to do is we're going to put a lot of stress with those phosphate groups, those negative phosphate groups on the end. It puts a lot of stress on this molecule right in the middle. And so now we're going to break it into 2 3-carbon molecules. Remember, each of them has a phosphate group on there. And then, for our next step, we are going to, and I'm just going to take 1 molecule, but remember there are 2. Alright, and I think I gave you guys the name for that molecule, and that is G3P, because we saw that in photosynthesis. So what we're going to do next is we are going to strip off some electrons. So if you remember in photosynthesis we were doing the opposite. We took in and we fixed carbon and we put electrons onto it. And in this chemical reaction we're going to take them off. Alright, so what we're going to do is we're going to bring in, what color did I have that? I think I had it orange. We're going to bring in NAD+, and we are going to pick up those electrons. So remember we talked about how we're going to reduce our NAD+ by picking up electrons. And if that molecule is being reduced, then of course we have our G3P, which is being oxidized, so it's giving up those highly energized electrons. The next molecule that we get from that particular chemical reaction, and I'm just going to put a 2 there because we have 2 of them, and I'm going to do both of them, is this. Here, go back to our little poop. And many of you guys should notice that, well, wait a minute, where did the phosphate group come from? Well, there's going to be some inorganic phosphate that's added up here. Right, so that again is going to be facilitated by enzymes. So we're going to add another phosphate to get our, you can say, your little poop. we're going to now take those phosphates off. This again. And of course, if remember, I showed you guys what those molecules actually look like, and you can see how they are different, but by just following the carbon skeleton, we're not seeing the changes in the oxygen and the hydrogens. So we are going to go from 2 phosphates down to 1. And again, we're going to actually make some ATP here. So we have to remember that we have 2 of these molecules. So how many ATPs did we actually make? We made 2 ADPs, we got 2. And now we're going to do this one more time. We're going to take that phosphate group off, and I'm going to have to kind of continue over here. Actually, I'm going to go like this. And pull the and remember, because we had 2 ADPs, 2 ATPs, and then we end up with our molecule called pyruvate, or pyruvic acid, depending on which naming system you go by. So what we basically have done here is we took our glucose, we have done some chemical reactions to harness the energy in order to make the energy transition from glucose to pyruvate easier to do by adding phosphates on. We stripped off some electrons. If we go up here, notice my numbers are not right because I have 2 molecules of G3P, so I need 2 NAD pluses. I get out 2 NADHs. To summarize what happened here, we put in glucose. Oh, that's not so right. Let's try that again. Going too fast in my head. We put in 1 molecule of glucose. We got out 2 pyruvates. We put in 2 ATPs. So we invested 2 ATPs. But we got 4 ATPs out. So let's say we profit. Well, I don't think let's say we profit. We got out. Our profit margin would be 2 ATPs. Is our profit. And we also ended up getting 2 NADHs, or we could say 2 full electron taxicabs that we referred to. And that is glycolysis.